Thank you everyone for joining. My name is Ana Maria, and I would like to welcome everyone to our René Salazar speaker series hosted by LatinX Abroad. I'm especially excited to be hosting today as we are going to start our Hispanic Heritage Month. And part of this initiative is part of our mission to build a thriving community among Latinos and allies in science, while also empowering the members of our community. We hope that through this program, we highlight the incredible work that our researchers are doing both here at abroad and beyond, as well as hopefully fostering collaborations between the speakers and people that are joining the talk, and also provide opportunity for networking. We hope that by showcasing these accomplishments of these individuals, we can provide a platform for people to learn about the research that Latinos are doing, and also try to show inspiring roles for underrepresented groups in science. Our goal here is to cultivate meaningful connections and promote groundbreaking research, as well as celebrate the richness and diversity of our community. So thank you again for joining us for this talk, and I hope you keep joining, and also please share the speaker series with everyone. From your network, we host this every month. So now I leave it to my colleague Ricardo, who will be introducing our speaker for today. Ruben, thank you for joining.
Thank you very much, Anna. So it's my pleasure to introduce Dr. Reuben Cordero. He's a postdoctoral scientist with Fisheries and Oceans Canada, and he explores aquatic ecosystems from Arctic freshwater habitats to coastal marine environments. He'll discuss how factors like glacial history, predation, aquaculture impacts species, and distribution and community interactions across these diverse ecosystems. This talk offers a glimpse into the complex dynamics shaping aquatic life, from historical climate events to modern-day environmental changes. Don't miss this opportunity, and take it away, Ruben.
All right, thank you very much. Let me see how this works. Okay, so first of all, I want to thank Ricardo and Ana Maria for this amazing invitation, and thank you all for attending this presentation named Exploring Aquatic Ecology, Insights from Freshwater to Marine Ecosystems. All right, so in this presentation, I will start briefly with my background, and then I will describe my work, starting with the work that I developed in my master's, followed by my PhD work, and then finishing with my current project as a postdoctoral research at Fisheries and Oceans Canada. Okay, so I'm from a city in Bogota, in Colombia, called Cúcuta, which is located at the border with Venezuela. And after, sorry, having some issues. Okay, then after high school, I moved to Bogota to study biology at the Universidad de los Andes, where I joined the lab of Professor Emilio Realpe. So in this lab, I did my undergrad and master's working at the Total Lake, which is a high mountain lake located at more than 3,000 meters of elevation. Here, I analyzed for my undergrad the levels of eutrophication by measuring phosphorus and nitrogen concentrations. And for my master's, I analyzed the levels of heavy metals in fish tissue, because this area is the largest producers of rainbow trout for human consumption. So eventually I moved to Canada, where I started a second master's at the University of Toronto, and the first analysis that I will present, which is called Geographical History of Aquatic Insects in Northern Canada. So this work was made under the supervision of Professor Douglas Corey. So the Arctic is one of the most fragile ecosystems in the planet, and is under great threat since the effects of the climate change are stronger in this region. Organisms in this ecosystem are highly adapted to extreme environmental conditions. However, they can be affected by fast changes of those conditions. And studies on species from Northern Canada are necessary to assess and compare how much impact climate change is having in this environment. So this motivated us, the Northern Biodiversity Program, which was an initiative between researchers from McGill University and the University of Toronto to document changes in the northern arthropod communities. So first, let's review the climatic history in North America and the impact of the glacial periods. During the last 1.6 million years, which is known as the Pleistocene, huge glacial advanced and retreated several times over North America. So here we can observe the global temperature fluctuation during the last 500,000 years and the multiple glacial and interglacial periods. The last glaciation period, which is called the Wisconsin, started about 75,000 years, and the last glacial maximum, which covered more than half of North America, occurred about 22,000 years. So this animation, this is an animation that shows how the ice retreated in North America since the last glacial maximum. And we see that initially the Beringia was exposed and ice-free, acting as a refugium from where organisms from bacteria to animals started to recolonize the new ice-free land. So we can observe that at the end, the Beringia land disappeared, and that's what separated North America from Asia. All right, so my study was focused on aquatic insects from the orders Ephemeroptera, Trichoptera, and Plecoptera for several reasons. For example, they are the base of the food chain in these aquatic environments. They are also widespread across northern Canada, including the Arctic archipelago. They are easy to collect and preserve, and they are sensitive to environmental changes, which makes them good indicators. So the objective of this study was to evaluate the influence of the last glaciation on intraspecific structure within these species. So we wanted to determine if there is evidence that widely distributed species of aquatic insects survived during the last glaciation in two or more refugia. For example, Beringia or the southeastern refugia. The hypothesis was that populations of widely distributed species that exhibit both genetic and geographical structure originated from two or more glacial refugia. So to collect the specimens, we went to these areas in northern Canada. We used standardized protocols of sampling on each site. And our team collected in 4 sites per ecoregion. That they were the Boreal, the Subarctic, and Arctic, to cover East and West Canada. For the specimens identification, I use a CO1 barcode approach, which is a very simple but efficient method to obtain a reliable genetic identification of a species. All the sequences were contrasted in the GenBank and Bolt database to obtain their identification, and then used to construct the phylogenetic trees of each group. But how we can differentiate species with genetic and geographic structure? Okay, so in this example, we can see that this mayfly species shows multiple individuals with a clear genetic structure, but not a geographical structure. While for this other species, we observe both the genetic and geographical structure, and the populations from Eastern Canada is different from the population from Western Canada. Okay, so for the results I obtained Bayesian phylogenetic trees for each order, Ephemeroptera, Trichoptera, and Plecoptera, and I could identify 18 species spread across East and West Canada. So from those species widespread, 7 species showed both the genetic and geographical structure, as this figure is showing. And in addition, we calculated the divergence time. And for 6 species, we observed a similar range of divergence times, suggesting a common event that matched with the duration of the last glacial period. While for only 1 species, that divergence time was earlier. And apart from that, the genetic distance between the East and West population of these species suggested instead that they were 2 different species, and that the glaciation generated this species event. So the main conclusions for this project were that I obtained evidence of the species with genetic and geographical structure. The divergence time allowed to detect the effect of the last glaciation on aquatic insect populations. We found evidence of the species occurring in different refugia during the last glacial period. And finally, I found also evidence of speciation due to glacial isolation. So for this work, I want to thank my supervisor, Dr. Curry, all the people in my committee, the Northern Biodiversity Program, and the people in my lab. Okay, so now I will talk about my PhD project at the University of Toronto, but this time with Professor Don Jackson. This project was called Parents of a Species Co-occurrence, a species pairwise approach. So the great ecologist Robert MacArthur stated that ecological patterns are only interesting if they are repeated. They might be repeated in space or in time, and they might be repeated from species to species. So as introduction, one central question in ecology is how natural communities are structured. And among the different approaches used to address this question, the species co-occurrence analysis have been a prevalent subject that goes back to the 1940s with classical studies that analyze co-occurrence patterns across multiple communities. However, it was the Assembly of a Species Community by Diamond in 1975, the publication that sparked and still ongoing debate about whether natural communities are structured by determined rules. In this publication, Diamond concluded that competition was the main mechanism structuring bird communities, which was in agreement with influential contemporary studies that also proposed competition as the most important mechanism structuring those communities. Diamond also mentioned that competition generates the forbidden combination, which means species that exhibit a pattern of segregation or negative co-occurrence. One example of this forbidden combination was observed between two species of cuckoo doves, which never co-occur in the same island as is observed in this map. So Diamond attributed this segregated pattern to the strong competition between those species for similar resources. However, Diamond's ideas were challenged by Connor and Simberloff in 1979, who argued that the effect of competition was typically assumed instead of being properly tested, and that the community should be analyzed entirely instead of selecting only the species pairs that exhibit these forbidden combinations. And for this, they proposed the null model as equivalent of the null hypothesis to test competition as main factor in natural environment. So the species co-occurrence analysis has been an active field since, and subsequent studies of species co-occurrence have produced important advances in this area, like development of robust metric or reliable randomization methods that allow to find evidence of non-random structure in natural communities. However, most of those studies have used the same approach that consisted in analyzing the identity of the entire community, ignoring individual species pairs association that can provide important ecological information. So we went from one side to the other. So for example, let's suppose that one pair of species 1 and 2, exhibit a large segregation value, while another pair of species, 3 and 4, have a large aggregation value. But when these values are added up, they can cancel each other out, producing a new co-occurrence value that is not different from random, hiding the actual ecological relevance of those species pairs. However, if we analyze individual species pairs, we can determine which pair are aggregated, segregated, or randomly associated, which increases the insight of the community structure and open questions about ecologically relevant species. So the general question of my thesis was, how does our understanding of community structure improve from the classical approach, the whole community, to the individual species pairwise approach? The objective was to test ecological hypotheses related to patterns of association between species, the signal of biotic interactions, and the influence of biotic and abiotic factors using a species pairwise approach. So I divided my thesis in 3 data chapters that I intend to go quick. So in chapter 1, I assess the community structure, consistency of association patterns, and community assembly theory using a species pairwise approach. In chapter 2, I aim to determine the influence of habitat size on these species association patterns. And in chapter 3, I wanted to determine the influence of biotic factors like trade on the species association patterns. So in the first chapter, I intended to determine whether and how species co-occurrence patterns are structured analyzing fish lake communities. This implied to determine their direction as well as their magnitude and consistency. In addition, I tested whether ecologically related group of species, like competitors, species that share habitat requirements, or predator-prey, exhibit co-occurrence patterns expected by community assembly theory. So for this chapter, I use the database of the Aquatic Habitat Inventory, which is a program of the Ontario Ministry of Natural Resources. So this database contained records of incidents of 99 fish species distributed in almost 10,000 lakes, grouping 144 tertiary watersheds, as is shown in this map of Ontario. So in this study, each watershed represented one community that was analyzed individually using a species pairwise null model approach to obtain the CS scores, which indicate the segregation level between each species pairs. With the obtained CS scores, I obtained also the standard effect sizes, which represented the co-occurrence value. And then for each species pair, I compared the standard effect sizes or the co-occurrence levels across all the watersheds they occur to determine how consistent was the pattern that they show. And in addition, I evaluated assembly hypotheses within ecologically related groups of species, competitors, predator-prey, and species with similar habitat requirements. Okay, so for the first part, the whole community, I analyzed a total of 76 fish species, and I obtained the association value for 78% of the possible species pairs. In this figure, the values of a standard effect size on the right side indicates a consistent segregated pattern or a species that co-occur less than expected by chance. The values on the left side indicate a consistent aggregated pattern or a species that co-occur more than expected by chance. And for species pairs with confidence intervals crossing zero, the pattern was considered not different from random. So overall, I detected consistent association patterns in 48% of the total observed species pairs, of which 27% were consistently segregated and 21% were consistently aggregated. So in the second part, the hypothesis of community assembly predicts association patterns expected for group of species biologically related. So among the processes expected to lead to segregation patterns, I focus on competition and predation. For example, it is expected that closely related species that compete stronger for similar resources exhibit a consistent pattern of segregation. However, if the association pattern is not consistent or is consistently aggregated instead, this could indicate that competition is not strong or other processes are more relevant. The same consistent segregated pattern is also expected for predator-prey species when predation is strong. However, other factors might also contribute to segregation patterns like habitat filtering or different geographic origins. And among the factors leading to aggregation patterns, I focus on the shared habitat requirements. So I analyzed the association patterns within the Centrarchidae family, which is composed by bases and some fishes. The members of these families are predators and occur in similar habitats, which according to assembly theory, should generate a pattern of segregation due to strong competition for similar resources. But instead, the results of the analysis between the smallmouth bass and other species of this family show consistent aggregation patterns. And the same pattern was observed between the other members of this family, which suggests that competition is not strong enough to generate segregation at this scale. Now, for the species with similar habitat preferences, I analyzed the association patterns of fish that require cold water. And the results show the expected consistent patterns of aggregation, indicating the strong effect of this mechanism on this group. Last, I analyzed the co-occurrence patterns between prey species like minnows and top predators of these systems like basses and northern pike. And I detected a consistent segregated pattern between each prey and predator. So these analyses, in addition, allowed me to determine what prey was more segregated by the predators. And we can observe that the stronger effect was observed on the northern red-bellied days. And besides, I could detect also which of the top predators generates the strong segregation effect on the prey, which was the northern pike. So the conclusions for chapter 1 were that the species pairwise approach provides an assessment of the overall community and allows testing specific patterns and their ecological relevance. This is because by pulling all the co-occurrence values, as in the classical whole community approach, result in a strong negative and a strong positive relationship canceling each other out, with the wrong conclusion that the community is structured not different from random. Also, species co-occurrence analysis can produce robust information about observed patterns that are repeated across multiple independent units, in our case, were the watersheds, in large scale geographic areas. And finally, at the scale considered in this study, I found evidence supporting predation and habitat preference as stronger mechanisms structuring fish communities than competition. Okay. So moving to chapter 2, here I wanted to determine the influence of habitat size on the co-occurrence patterns. So among the environmental factors, area, and specifically the species-area relationship has been considered one of the few laws in ecology, and it has been one of the longest analyzed and most frequently demonstrated in nature. So here we see a classic example of this pattern detected by MacArthur and Wilson on Caribbean island, where the richness of amphibians and reptiles increases with island area. Studies evaluating the influence of area on specific on species richness has also been applied to lakes. This is because lakes are considered analogous ecosystem to islands due to first, the clear area delimitation as is observed in islands in the Bismarck Archipelago or lakes in Ontario. And second, elevation in islands is related to habitat diversity, which allows, for example, in plants, the establishment of different communities along an altitudinal gradient. This factor is analogous to depth in lakes, which generates habitats with different characteristics that allow different types of communities. So area and depth influence the distribution and richness of a species and has been tested several times. However, it is still not clear how these factors can influence the co-occurrence patterns in fish communities. So the general goal of this chapter was to determine whether and how abiotic factors related to habitat size like lake area or depth influence the species co-occurrence patterns. So to determine the effect of habitat size on the species co-occurrence, I grouped adjacent tertiary watersheds with similar species composition in 2 different regions in Ontario. This allowed me to increase the number of lakes to gain more statistical power because we analyzed more than 1,100 lakes per region, still controlling for biogeographical influences. So I will skip the methods here, and we can talk later about them, but basically I use a randomization test to determine significance between lake area and depth categories and the individual species pairs co-occurrence values. So in the second part, I wanted to evaluate the effect of area and depth in group of species that potentially interact like predator-prey or species with similar habitat requirements like cold water fish. And for this, I use a meta-analytical approach to determine the whole effect between one single species with the other species in the group. So in the results across all species pairs, I found that both factors, area and depth in both regions, central and western, exhibited highly significant differences between the observed and expected distribution of census slope, indicating a strong influence of habitat size on the co-occurrence patterns. Now, for the group of predator-prey, I analyzed the co-occurrence values between each predator and each prey, and I detected significant differences between the categories, which indicates the strong influence of lake area in this group. So in this forest plot, we can observe that the strong negative co-occurrence effect in the smaller lakes, okay, lakes with less than 25 hectares, is significantly decreases in larger lakes, indicating that species segregated in smaller lakes can coexist in larger lakes. Now, for the group of cold water species, I found no significant differences in co-occurrence values across the depth categories. However, we can observe a tendency for these species to be more aggregated in deeper lakes. So in addition, we observe the lack of association patterns in the smaller category, which indicates the strong environmental filtering for these species in shallower and warmer lakes. So these results suggest that when lakes reach a minimum required depth, these species tend to co-occur positively. So the main conclusions for chapter 2 were that habitat size does exert a significant influence on the species co-occurrence patterns. In the group of predator-prey species, segregation effect was reduced when area increased. And it is important to recognize how habitat characteristics reveal ecologically relevant information that might be concealed when these variables are not considered. Now, chapter 3 was about the influence of species traits on co-occurrence patterns. So biotic interactions have been recognized as important factors structuring communities, and the magnitude of those interactions can be related to the degree of similarity of species in their traits. The trait-based approach have been employed to predict community structure by assessing whether the patterns observed can be explained by the proposed mechanisms. For example, species with similar traits are expected to compete stronger due to more resource overlap than species with less similar traits. So among the species traits, body size have been recognized as major influence on the strength of a species interaction, particularly in aquatic ecosystems, as the structure of this community can be determined by the prey size range that a predator is capable of consuming. Another important trait is the temperature preference, which has been observed to influence the community structure by selecting or restricting species from various locations. For example, plant communities adapted to low mountains versus communities adapted to high mountains, or cold versus warm water fish assemblages. Trophic level is another important trait that has been used to define the position of species in the food chain, and it can be used as well to identify potential competitors or species involved in predator-prey interactions. So in this chapter, I intended to determine whether the mentioned traits can explain the co-occurrence patterns obtained for all the species pairs and whether the signal of biotic interaction can be detected. Okay, so I intended in this chapter to test 4 hypotheses. Don't worry, I'm gonna go quick about this. So the first hypothesis is that across all the community, or in general, the trait values are significantly related to the co-occurrence patterns across all the species pairs. Hypothesis 2 is for body size, and I expected a negative and significant relationship between body size dissimilarity and co-occurrence patterns if the signal of competition is strong. This is because species with similar body size tend to compete stronger and segregate each other. But the segregation level can be reduced when differences in body size increase. Or alternatively, a positive and significant relationship if the signal of predation is strong, and this is because species with similar different body size tend to have a predator-prey relationship and segregate each other. Hypothesis 3 is about temperature preference, and for this trait, I expected that species pairs having more dissimilar temperature preferences exhibit more segregated patterns due to environmental filtering, while aggregation is expected for species with similar temperature preferences. And for hypothesis 4 about trophic level, very similar to body size, I expected a negative and significant relationship between trophic level dissimilarity and co-occurrence patterns if the signal of competition is strong, because species with similar trophic levels tend to compete stronger and segregate each other. And the segregation level can be reduced when differences in trophic level increase, or alternatively, a positive and significant relationship if the signal of predation is strong, because species with different trophic levels tend to have a predator-prey relationship and segregate each other. All right, again, so to summarize the methods, I performed a series of multiple regressions and permutation tests between the co-occurrence values and the traits to determine the significant effects. We can talk about this later if you have some questions. So the result for hypothesis 1, including all the traits and the randomization tests for all the species pairs, show significant relationships between the co-occurrence value with temperature preference and trophic level, but not with body size. For hypothesis 2, the result for body size dissimilarity at co-occurrence values shows a negative quadratic trend, although this relationship was not significant according to the permutation test, indicating that body size dissimilarity do not predict values of co-occurrence between species. For hypothesis 3, we observe a positive and significant linear trend between temperature preference and co-occurrence values, indicating the strong influence of environmental filtering. That means that species with larger differences in temperature preference tend to be segregated. And finally, for hypothesis 4, I obtain a positive and significant relationship for both the linear and the quadratic term of trophic level and the co-occurrence values, which suggests that species with larger differences in trophic level tend to co-occur negatively. However, here the signal of predation is observed, while the signal for competition was not clear, corroborating previous results. All right, so the main conclusion of chapter 3, I detected significant association patterns between the predictor and the response variables, which indicates an overall influence of the traits on the co-occurrence patterns. Temperature preference was highly associated with the co-occurrence values, which indicate a strong influence of environmental filtering in the structure of these communities. Trophic level was significantly influences the co-occurrence values, and species with different trophic levels tend to segregate each other. The signal of predation was detected here, but not the signal of competition. And finally, contrary to expected by theory, body size was not significantly related to the co-occurrence pattern, so this trait cannot predict interaction between species. So in this part, I want to acknowledge, I want to thank my supervisor, Don Jackson, and the members of my committee, and all the people from Jackson Lab, and yeah, like all the people that made possible this journey. And last, I will present the current project that I'm working on as a postdoctoral researcher at the DFO, Department of Fisheries and Oceans, Canada, about the effect of shellfish aquaculture industry on zooplankton size distribution in several coastal embayments in Canada.
Hey, Ruben, would you like to take a question about the last section, or would you like to keep them for
yes, if, I don't know.
So we have a question from Erwin Jungeries. If competition wasn't a big factor, why did competing species consistently aggregate rather than just being random?
I mean, we need to understand that I am analyzing at the level of the lake, right? Within the lakes, there are many habitats that they can where competition can be important. We need like a more experimental, and these experiments have been performed for like years, showing that competition is very important. But at this level, competition didn't show like a clear signal, while predation did. So that's the main difference. It's not that competition is not acting. It's not acting at the level that I analyze. Does it make sense?
Yeah, I believe so. I didn't have a follow-up, or I didn't see a follow-up question.
Okay, we can talk about a little bit later, but
yeah, for sure.
That's the main reason. It's just the scale that I was analyzing. So again, so the shellfish aquaculture in Canada is an industry that has experienced substantial growth since the 1970s, and today is a thriving industry producing millions of metric tons of food, meeting not only domestic but international demand, and expanding across coastal zones, highlighting its economic importance. However, as this industry expands, the focus of ecological studies has increasingly shifted toward understanding of the interaction of this activity with surrounding habitats, specifically analyzing the effect of higher trophic levels and local planktonic communities. So shellfish feed mainly on planktonic organisms, and the high density of shellfish produced by the aquaculture activity generates different effects on the local habitat and organisms. One major effect is the grazing pressure generated by the shellfish on higher trophic levels. For example, experimental studies found a significant effect of the shellfish density on the development of lobster larval stages, and because the strong competition exerted by shellfish on the microplankton resource, lobster larvae exposed to higher shellfish density exhibited lower development and survival. But how is the planktonic community structured? So basically, the plankton is composed by two main groups, the phytoplankton and the zooplankton. Phytoplankton is composed by the primary producers, the organisms transforming and storing the solar energy, accounting for most of the smaller size fractions like the pico and nanoplankton. And studies that analyze the impact of shellfish grazing pressure on this group observe that the smaller phytoplankton fractions that cannot be filtered by shellfish exhibit an increment in abundance compared to the larger fractions more intensely grazed. The other group is the zooplankton, and these organisms are the main component of the larger size fraction of plankton, like the mesozooplankton, which is the focus group of this study. So let's briefly review the importance of this group. The mesozooplankton fraction has two main key characteristics that makes it an interesting study system. First, it plays a fundamental role in marine ecosystem by acting as a critical trophic link, facilitating the energy transfer from the primary producers or phytoplankton to higher trophic levels that includes other bivalves and other fisheries target species. And second, zooplankton exhibit a high degree of environmental sensitivity due to their physiological adaptations and life history strategies, making them highly responsive to variation of environment conditions like temperature, salinity, and nutrient concentrations. So given these attributes, many researchers have advocated for the establishment of enhanced monitoring programs to facilitate the early detection of ecological impacts, especially in our current scenario of climate change. But although several large-scale programs have been established to monitor long-term changes in marine health, there is still a scarcity of monitoring programs specifically designed to track the continuous effect of aquaculture on planktonic communities. So this motivated the initiative of the Aquaculture Monitoring Program by the DFO, which aims to track and analyze changes in the structure of planktonic communities to develop strategies and conservation policies. So we have seen that the mesozooplankton plays a key ecological role as link between primary producers and higher trophic levels and is highly responsive to environmental changes. As well, we know that shellfish aquaculture has a range of effects on these planktonic communities, and although these effects have been evidenced by theoretical and experimental analysis, still significant gaps remain, specifically concerning the long-term consequences of this interaction in natural environments, both within and beyond bay-scale contexts. So then the goal of the Aquaculture Monitoring Program is focused on the long-term sustainability of the shellfish aquaculture industry. The general objective of the AMP is to track changes in the coastal ecosystem by observing alteration in the structure of zooplankton communities in presence of shellfish aquaculture and provide managers with robust and reliable data to make more effective conservation policies. Specifically, we focus on determining the level of variation in the mesozooplankton size structure generated by three factors: the station or location within the bay, which is related to the morphological characteristics of each bay; tide level, which is related to the replacement of grazed individuals by water exchange; and the day of sampling, which is associated with stochastic events like rain or wind. In addition, we also aim to determine the seasonal variation pattern in the mesozooplankton size fraction by comparing monthly samples associated with the local water temperature bay. So to address this objective, the AMP team collected plankton samples in different bays with presence of shellfish aquaculture across several regions, including Newfoundland, the Maritimes, the Gulf of St. Lawrence, and the Pacific, as is observed in the map. At each bay, the goal was to sample in 3 different locations or stations for 3 consecutive days, and each day there was one sampling during high tide and another sampling during low tide. So these samplings were collected during the summers of 2020 and 2021. And in addition, monthly samples were obtained at South Arm Bay in Newfoundland from June 2021 to July 2022. After the samples were obtained and preserved, we used the macroflow CAM methodology, which generates and measures microscopic individual images to obtain 18 parameters related to body size for each collected individual. So we selected the biovolume prolate spheroid, we're going to call it biovolume, because this parameter accounts for the organism's shapes and surface area in relation to its volume and for comparative purpose with recent studies. So the AMP team aimed to collect and analyze plankton individuals with sizes above 30 micrometers, but for this analysis, I include only the individuals of the largest fraction size between 250 and 2 millimeters. Okay. So in the results, we obtained more than 500,000 individuals biovolume values across the study base, and we generated these volume plots to visually detect differences in the biovolume distribution of mesozooplankton across the different bays. This figure shows the different size distribution across the analyzed bays, indicating that bays are not homogeneous in their zooplankton size structure. So to quantify the biovolume variation, we obtained the cumulative frequency distribution of each samples, and they were compared pairwise to calculate the maximum variation between those samples associated with the analyzed factors. For example, this figure shows two cumulative frequencies from two samplings, comparing the effect of tide. So one sampling was made in high tide and the other one was made in low tide, and we compared that, and exhibit low variation. And we quantify that variation by calculating the maximum distance using a Kolmogorov-Smirnov test, which in this case shows about 7% of variation between these samples, indicating a low effect of tide. While for these two samples in the right, we see a greater maximum distance of 35% between those samples, indicating a greater effect of these factors. Then the maximum distance from all the comparisons related to each factor, tide, day, and station, were pooled to obtain the variation generated by each factor at each bay. So here we can see the result for the station effect, and in general, we observe a low biovolume variation across the bays, with the exception of St. Peter's, where the variation between stations shows a median of 23%, indicating a potential effect of this factor in this bay. Similar results were observed for the tide effect, with low values of variation detected for most bay. As well, we can see here the higher variation in St. Peter's Bay with around 16%, but still indicating a weak effect of this factor in the biovolume variation. And the factor day showed similar results than the previous factor, but the variation level associated to this factor was much larger for St. Peter's than the other bays, with around 35% of variation when comparing samples from different days. Finally, we pulled together all the variation from the three factors, and we observed the overall small biovolume variation across the compared bays, as well with St. Peter's showing the largest overall variation. A potential explanation to this variation in St. Peter's might be related to the higher proportion of area used for shellfish aquaculture in this bay, which is around 40% of lease coverage compared to the other bays. However, we are still testing this hypothesis. So now we focus on St. Peter's, which is the bay with higher variation, and we determine the amount of variation between the categories of each factor. And this figure shows that the factor day, for example, the higher variation was observed between day 1 with days 2 and 3. And for the factor station, the largest differences were observed between the outer station with the mid and the inner stations, and the level of variation due to tide effect was lower than the other factors. So we also generated this NMDS, which is a non-metric multidimensional scaling. It's an ordination plot. This plot allows us to see a separated group of samples from the outer station, grouped by the red dash ellipse, apart from the other stations, and also we observe the green ellipse, the samples from day 1, group apart from the other days. Now, we wanted to test the significant factor effects, and for this, we perform linear mixed effect models, comparing 3 quantiles of each sample distribution with station and tide as the fixed effect, and day considered as the random effects. So the results show significant differences for the station across the 3 quantiles between the outer and the middle stations, and between the outer and inner station, we observe significant differences at the larger biovolume quantile. For the random effect day, the model results show highly significant differences across the 3 quantile, indicating a strong effect of this factor representing stochastic events. The factor tide and its interaction with the station show no significance. So finally, to determine the seasonal effect, we analyzed monthly samples collected between June 2021 and July 2022. And we obtained the biovolume fraction distribution for 13 months. And as expected, we observed significant differences in the fraction distribution across different months. So in general, we can see a subtle pattern where the largest biovolume media values were associated to the colder months. The temperature trend can be tracked with the red dashed line and the temperature values in the right y-axis. And finally, our regression analysis between the median biovolume and the mean temperature show a negative significant relationship supporting this pattern. So the main conclusions of this work are that in general, we observe a low variation in the volume fractions distribution between samples related to the factors analyzed, with a higher or like an exception observed in St. Peter's. Contrary to expected, the factor day of sampling, which we thought that it was not like important, related to stochastic events, show more variation than the tide level or the station. The higher variation observed in St. Peter's might be potentially related to the more intense shellfish aquaculture developed in this bay. The seasonal pattern showed that colder months exhibit greater abundance of larger mesozooplankton individuals. And as a general conclusion, long-term monitoring programs are fundamental to track changes in the size fraction structure of planktonic communities, which is the food of the shellfish industry, to ensure the sustainability of this growing and necessary activity. So with this, I want to thank the PIs of this project, also the members of the Data Lab here at Dalhousie University, to all the members and collaborators of the AMP team. Again, thank you to Ana Maria and Ricardo for this opportunity, and thank you all for your attention, and I'll be happy to take any questions.
Awesome. So thank you very much, Ruben. So first thing is that we have a short survey. So if you can, please fill out the survey because your feedback is very valuable. And we're going to open the floor for questions. So Erwin also had another question, but why aggregate rather than being random or negative? And that was from the end of the first section. I don't know, Russell, can we get Erwin on a mic? Maybe he wants to have more of a discussion. Up to Erwin. But if you can start by answering the question, and Erwin can let us know. Oh, there he is. Yeah.
Hello, can you hear me?
Yes.
Yeah, I'm referring to a slide that was right at the end of the first section where you showed the competing species tending to aggregate.
This one's?
I can't see your slides, sorry.
Sorry, yeah.
Right, so if the species were competing, the original hypothesis was that they would segregate. And I understand you said that at the level you're looking, that was not strong enough to detect. But if that's so, I guess I would expect that what you'd see is sort of random, whereas what you're seeing is consistent aggregation rather than neither. So why are they consistently aggregating?
Well, they are still like all these species, they share similar habitat requirements. So in the paper, we connected these ideas to like saying that this factor was stronger than competition organizing the structure of these communities at this level, right?
Okay.
So if we go deep inside the lake, we are going to see that areas where like I don't know, the small mold bass is the largest of these centracids, were like a present like in like around that area in the lake, within the lake. There is not going to be any other centracid, especially the small ones. But we are like just checking the presence absence across in the entire lake. So that's the main reason why competition was not observed, the signal.
Okay, thank you.
Yeah. So please feel free to pose questions. So I had a question actually. So in your first section, you said there's like an 8% difference between the species. Is that like, what is, like, how do you measure, is there a cutoff for species divergence? Or is there?
According to the CO1 papers since the beginning, their agreement was like around 2%, and that's based using like a molecular and also morphological data. So when a species is about 4 or 5%, it's considered two different species. When populations show that amount of genetic divergence, they are considered two different species. So in our case, we observed like all the species were below that threshold. Here, for example, this species is less than 2, or like around 2. This is way less than 1. But here we can observe the genetic distance is 3 plus 4. That's almost 8%. So yeah, that started to indicate that possibly it's the two different species. And the speciation event was because they were separated in the Beringia and the southern plains refugia.
So it's have you seen indication of not just like geographical distribution that changes speciation, but maybe like they go to different like niches, ecological niches?
That was, I mean, like I'm pretty sure like there should be like evidence for that, but that I didn't delve into that part. So we're just focusing on this big scale.
And have you seen a lot of molecular changes without many like morphological changes, or is it both have to be a coincidence to define a species?
I mean, like this species is still considered one species in North America. So I don't know if if there is like a taxonomy in this group. I invited to dig into this part because it's very interesting how these species, they look similar. They are occurring in similar habitats. So the differences are not very clear. So yeah.
Yeah, so I'm just going to ask one more time. If anyone else has questions, please feel free to ask. Otherwise, we're coming up against the hour. So once again, I'd like to thank Ruben for doing this talk for us. We had a lot of interest from a lot of different types of people at the Bro, which is really cool. So we had some people who were super interested in evolution. So that was great. So thanks again. And we will end it.
Okay. Thank you, everyone.